Good morning and welcome to all. So in last session we have discussed up to the detailed structure of endospores. We have discussed the outer proteinaceous coat, the cortex and a germ cell wall. Next we can discuss about inner membrane. Under the germ cell wall, it is the major permeability barrier against several potentially damaging chemicals. It can be this inner membrane which can be similar to the plasma membrane. Next is about the core, which is the center of the endospore. It exists in very dehydrated state and it houses the cells, DNA, ribosomes and large amount of dipicolinic acid. This endospore specific chemical can comprises up to 10% of the spore's dry weight and appears to play a major role in maintaining spore dormancy. So it has a major role in maintaining the dormant structure of the spore. Next is about small acid soluble proteins or SASPs, which are also only found in endospores, and these proteins tightly bind and condense the DNA. So these are present in the core portion and are the part responsible for providing resistance to the UV light and DNA damaging chemicals. So it protects the DNA from the damaging chemicals by tightly forming a bond or covering of a small acid soluble protein. Next is other specific structures and chemicals which are associated with this endospores include stalks, toxin crystals and also an additional outer glycoprotein layer which is known as exosporium. These structures also provide resistant property to the endospores. Next is about endospore development. The process of forming an endospore is very complex and we usually have selected a model organism for studying this endospore formation, and it is Bacillus subtilis. It is a gram-positive rod-shaped bacteria. Endospore development, it requires several hours to complete. It is not an easy process. So during this process, key morphological changes occurs and it has been used as a marker to define the stages of development. So several different types of morphological changes occurs during the endospore development. As a cell begins the process of forming an endospore, it divides asymmetrically. So first of all, the cell division which may occur in asymmetric fashion. This results in what? A creation of compartments. That is, it consists of a large mother cell and a small forespore. So first of all, cell division occurs in asymmetric pattern, a large mother cell and a small forespore. These two cells have different developmental phases. So these two cells have different fate in their development. Intercellular communication systems which coordinate cell-specific gene expression. So there is a communication that is called intercellular communication system which coordinate cell-specific gene expression through the sequential activation of specialized sigma factors in each of the cells. The next stage is stage 3. In this, the peptidoglycan in the septum is degraded and the forespore, it is engulfed by a mother cell, forming a cell within a cell. So the peptidoglycan in the septum, it is degraded. So what happens? This peptidoglycan is degraded and a forespore. Now the forespore, it is now engulfed by the mother cell, which have been forming a cell within a cell. So it looks like a cell within a cell. the activities of the mother cell and the phosphore, which leads to the synthesis of the endospore specific compounds. So, endospore specific compounds are synthesized due to the activities of the mother cell and phosphore, and it leads to the formation of the fourth and fifth stage, which are known as cortex and deposition of the coat. Finally, it is followed by the dehydration and maturation of endospores, which indicates sixth and seventh stage. Finally, the mother cell is destroyed in a programmed cell death. So, the remaining what happens? The phosphore converted into an endospore and it is released into the environment. This is the steps in the formation of endospores. The endospores will remain dormant until it senses the return of more favorable condition. If the favorable conditions are met by this endospores, it converted from the dormant to the active stage. A sigma factor, it is a small protein that directs the RNA polymerase to specific sites on the DNA to initiate gene expression. For the conversion of the dormant to active stage, there is required the action of there is a need of initiating a gene expression, and this gene expression can be initiated by the sigma factor, which directs this RNA polymerase. This is the stage which I have discussed. Stage 1, that is asymmetric cell division forming a septa, and this is the mother cell and phosphore. And next is the other stages of the spore, cortex and coat synthesis. Finally, spore maturation and mother cell lysis. So, we have already discussed the reactivation, that is any favorable conditions are met. When the favorable conditions met, what happens? The spore germination occurs. So, this stage is reactivation. The reactivation of this endospores occurs when the conditions are more favorable, which involves activation, germination and outgrowth of the endospore. Even if an endospore is located in plentiful nutrients, it may fail to germinate unless activation has taken place. So, activation is most important. Only then the endospore germinates. Then this can be triggered by what? Heating the endospore. That is the process called heating the endospores. The next is germination involves the dormant endospore starting its metabolic activity. Now it becomes active by the metabolic process taking place inside the bacteria, inside the endospore, and thus breaking its hibernation. It is commonly characterized either by rupture or by the absorption of spore coat. Then swelling of the endospore occurs, an increase in the metabolic activity occurs, and it may result in the loss of resistance to the environmental stress. Now it is germinated and reactivated. Endospores possess 5 times more sulfur than vegetative cells, and this excess sulfur is concentrated in the spore coat, that is outer coat, as an amino acid cysteine. So, the sulfur is accumulated in the spore coat in the form of amino acid cysteine. It is believed that the macromolecule accountable for maintaining this dormant state has a protein coat rich in cysteine, which is stabilized by SS, that is disulfide linkages. So, the dormant state has a protein coat rich in disulfide linkage, which is an amino acid. The reduction in these linkages has the potential to change the tertiary structure, which may cause, whenever the tertiary structure is changed, the protein is unfolded. So, there will be a conformational change occurs in the protein. and this can be thought to be responsible for active enzymatic sites, exposing those active enzymatic sites necessary for this endospore germination. So it might be the reason for the endospore germination by reduction of this linkages. Next is importance of the endospores. As a simplified model for cell differentiation, the molecular details of endospore formation has been extensively studied, specifically in the model organism, that is Bacillus subtilis. We usually selected this organism. The studies have contributed much to our understanding of what regulation of the gene expression, transcription factors and sigma factors subunit of RNA polymerase. So we have studied several different factors for the endospore formation as well as germination in the organism Bacillus subtilis. The endospores, then another importance is that the endospores of the bacterium Bacillus anthracis were used in 2001 anthrax attacks. So this, in the 2001 anthrax attacks, the endospores of this Bacillus anthracis were used. The powder found in the contaminated postal letters, it was in the form of a postal letter which was contaminated with by seeing a powder, and that powder consists of anthrax endospores. This intentional distribution led to 22 known cases of anthrax. Among the 22 known cases, 11 inhalation and 11 cutaneous rates. The case fatality rate among those patients with inhalation anthrax was 45%, that is 5 among 11. The 6 other individuals with inhalation anthrax and all the individuals with cutaneous anthrax has also been recovered. Had it not been for an antibiotic therapy, many more might have been stricken. According to the WHO veterinary documents, Bacillus anthracis sporulates when it sees oxygen instead of carbon dioxide present in the mammal blood. This signals to the bacteria that it has reached the end of the animal and an inactive dispersible morphology is useful. Then the sporulation requires what? The presence of free oxygen. In the natural situation, this means the vegetative cycles occur within the low oxygen environment. The vegetative cycles occurs within the low oxygen environment of the infected host, and within the host, the organism is exclusively in the vegetative form. Once outside the host, the sporulation commences upon exposure to the air, and the spore forms are essentially exclusive phase. Next is about the important application in the biotechnology. This Bacillus subtilis spores are useful for the expression of recombinant proteins. And in particular for the surface display of peptides and proteins as a tool for fundamental and applied research in the field of microbiology, biotechnology and also in the vaccination. So this Bacillus subtilis spores are useful for the expression of this recombinant proteins.